So, to our first speaker, Dr. Ian Pringle. Ian is a postdoctoral scientist and has worked at the UK CF Gene Therapy Consortium for a number of years. He has a BSc in Applied Molecular Biology from the University of Aberdeen and a higher degree in biochemistry from Trinity College in Oxford. As a molecular biologist, Ian's main focus in the consortium has been the development of gene therapy vectors. He had a leading role in the creation of the consortium's current clinical trial product, the GL67A/PGM169. He made the PGM169, and hopefully he'll tell us what that all means. And today he continues to develop novel vectors. He's a previous winner of the British Society for Gene and Cell Therapy's Outstanding Young Scientist Award, and so it's our privilege to welcome him today to demystify cystic fibrosis genes and genetics. Ian, let me get your presentation.
Okay, thanks very much, Matt. I'd like to thank you for the invitation to come here today. It's a great privilege to be involved in this.
Sorry, that's the wrong. That's the slide I said I wouldn't show.
Yeah, skills, excellent. So yes. Yes, good afternoon. My name is Ian Pringle. I'm a scientist with the consortium. If you want some proof of that, here's me being a scientist quite recently and staring at something in the way that only scientists really can. What do I do for the consortium? To be honest, I don't really have a job title or a really tight job description. Very few of us really do. My aim really is just to try and advance or support our research wherever I think I can. As Matt said, I am involved heavily in gene therapy vector design. I do a lot of molecular biology work for that. And I also do molecular assay development, similar to what people are familiar with in terms of screening for CF mutations. I work a lot with robots and try to automate a lot of our lab work. I run a number of animal studies on our mouse models, and I'm heavily involved in software tools, database and website development to support the consortium's work. But today I'm here to talk about CF genetics. But first of all, I just want to talk a little bit about the consortium. I've been with the consortium since it started, and it has always been a tremendous challenge, but the work we do I think is important, and I'm so proud to be part of this fantastic organisation working with Uta in London and the guys up in Edinburgh. So before I do anything else, I just want to start by acknowledging everyone in the consortium and all of their continuing efforts in our progress. So thanks to everyone in the consortium. Today I'm here to talk about CF genes and genetics. I'll say at the outset, I'm not a massive expert in genetics. I thought I was, but it's one of these things which has improved a lot over the years, and it's advanced massively. So I'm going to talk a little bit about some background, and then I'm going to look at the genetics of cystic fibrosis. I'm going to give an example of how it affects us in the lab, and then by way of a conclusion, I'm just going to look a little bit into the future. The structure of DNA was first uncovered by James Watson and Francis Crick back 60 years ago in 1953. And in the opening line of their Nature paper, they have this phenomenal understatement where they claim that the structure has novel features which are of considerable biological interest. And they were certainly correct. The structure of DNA is both incredibly complicated and at the same time beautifully simple. It's comprised of just 4 simple nucleoside bases, A, C, T and G, and these are connected by an incredibly strong sugar phosphate backbone to produce long strands of DNA. And then we have complementary base pairing between the 2 strands of DNA to provide an incredibly strong rotating helical structure that people are used to seeing in models and illustrated here in this diagram. The structure, I couldn't go back. The structure of DNA was, once it was uncovered, it was immediately obvious that it had a major role in passing on genetic material from one generation to another, and it was obvious as well that it had to be involved in coding for proteins. But it wasn't immediately obvious how this worked. And it wasn't until much later, when in 1961, again, Francis Crick, now working with Sidney Brenner, discovered the amino acid triplet code. And what was discovered was that DNA is copied, or transcribed, into a secondary molecule, messenger RNA. This RNA is then translated, and a protein molecule is then produced. And the work they did in 1961 uncovered that it takes 3 DNA bases, or a codon, to code for 1 amino acid, and amino acids are the building blocks of proteins. So the so-called amino acid triplet code was uncovered, and what we have is a system whereby an individual triplet of DNA codes for just one amino acid, but many amino acids are coded for by multiple different codons. And this code, which is presented here, taking simple DNA sequences and converting it to amino acids, is almost entirely universal across all forms of life. One gene, the simple unit which codes for a protein, one gene codes for roughly one protein, but there may be one, but particularly in higher animals and humans and other mammals, multiple versions of proteins can be coded for by an individual gene. And genes can code for hundreds or maybe thousands of amino acids, so the variation in size is quite striking. Every human cell has the exact same number of genes, the exact same genes indeed, whether it's a nerve cell, a photoreceptor, or macrophage, or anything. And prior to the Human Genome Project, it was estimated that we had over 100,000 genes. It turns out we've got many, many fewer than that, just over 30,000. And a mutation in just one of these genes, the cystic fibrosis transmembrane conductance regulator gene, causes cystic fibrosis. But because every cell has the exact same genes, you can take a DNA sample from blood, which isn't affected by CF, and screen for CF mutations. Our DNA is arranged on 23 chromosomes, and in total we have roughly 3 billion base pairs of DNA spread over these chromosomes. Only 2% of our DNA actually codes for proteins. And it used to always be said in textbooks for genetics that this meant that 98% of our DNA was what was called junk DNA. I think nowadays we move, with greater understanding that we have of our genome, a more polite or more accurate description, in fact, would be to say that 98% of the DNA is required to regulate the 2% that does the work. And so it really is not that the notion of junk DNA really isn't something that we consider these days. Some other headlines about our human genome. On average, any two humans differ only once in 1,500 bases. This still means that we have in the region of 1.4 million common mutations that can be mapped to individual chromosomes across the genome. In truth, men have slightly more mutations than women, and so here's proof if it really was needed, what women have always known, that we do in fact make more mistakes. Everyone has the same genes in the exact same order, and there's no genetic basis for race. Amongst this sea of conservation and similarity, when you see a variation, it stands out quite easily. And the ability of, the presence of variation standing out is how we're able to find mutations and map them and understand what they do. Because when mutations occur, they can be easily identified. Nowadays, this is generally done through direct sequencing of DNA, and you can see an example I drew here. And once you've identified a sequence, it's relatively easy then to design sequence-specific assays, and there's many ways of doing this, that will specifically detect either the mutated sequence or the wild-type sequence. And there are many systems available nowadays that can screen for hundreds of mutations at a time, and this is the basis of diagnostic tests for cystic fibrosis or other diseases. And it's also the way in which we do a lot of our molecular assays in the lab. The CFTR gene is located on chromosome 7. We've known this since the mid-1980s, and back in those days it was much harder to locate the location of a gene and understand what it did. The total sequence of the CFTR gene is around 250,000 base pairs, but only the sections which I hope you can see in red here code for the protein. It's a reasonably large protein for humans. It's about 1,480 amino acids long. And one of the perhaps the most remarkable thing about the CFTR gene is that in truth it is a completely unremarkable sequence. It's what's known as a housekeeping gene. It's expressed at very, very low levels in airway cells. Some estimates I've made from our own work is that in nasal epithelia it can be expressed as low as 1 to 2 copies of messenger RNA per cell. Phenomenally low levels for a steady-state protein. But there are over 1,900 mutations that can lead to cystic fibrosis. So why are there so many mutations in CFTR? 1 in 25 Caucasians are carriers, but don't suffer from cystic fibrosis. So it's been long felt that there must be some evolutionary advantage to being a carrier. And the most compelling theory for this is that carriers were, in our past, more resistant to diarrhea. And this work is backed up by some studies done in the early 1990s on cystic fibrosis CFTR knockout mice, and it was found that mice that were carriers for CF mutations, that didn't have carriers for that had one copy of the CFTR mutant gene, had lower CFTR activity in their intestines, and this meant that when they were exposed to an intestinal infection, they secreted less water in response to the infection. Therefore, the theory is that medieval European carriers of CFTR were less likely to die from dehydration. It's a very clean, very nice theory. In truth, though, there's actually less evidence of this really playing out in humans. Carriers seem to have normal CFTR activity, CFTR chloride activity in the intestines, and the frequency of CF, 1 in 25 carriers in our population, is still a little bit too high to map to the late emergence of cholera epidemics in Europe. So the likelihood is that this kind of diarrhea resistance theory is involved in the high incidence of CF in some way, but it's still the most likely explanation, but it's also likely that there are other factors that are involved in its prevalence as well. It's important to note that we're talking here about human evolution playing out over thousands and thousands of years. So founder effects have time to be very subtle and have time to be built up over that time. And the definitive experiment, which we will never be allowed to do obviously, is to take 1,000 carriers of CF, 1,000 carriers and 1,000 non-carriers, give them all cholera, and see what happens. I wouldn't volunteer for that, and health and safety these days. But why are mutations of this gene such an issue? Well, CFTR is a real keystone protein in wherever it's expressed. It's a very complex transmembrane protein, as shown by this diagram here, and so the folding and the structure that this protein has to take when it's active is really, really crucial. CFTR regulates chloride transport across epithelia, and normal CFTR controls the movement of controls chloride concentration between the inside and the outside of cells. Whereas mutant CFTR, in the case of that, there's no movement of chloride, and you then have a build-up of mucus on the outside of cells, causing CF lung disease. Is CFTR simply a chloride channel? No, it seems to move other ions as well. There's a lot of interest these days in its role in transporting bicarbonate. And there's also evidence that it's involved in the killing of Pseudomonas as well. Good studies done on that. It's also known to interact with many other proteins in the cell, and so loss of CFTR function will influence the function of these proteins too. And the truth is that no one really understands completely the roles of CFTR. If you think you do, you simply haven't realised how complicated it is yet. Mutations fall into 6 different classes, although I'm only really showing 5 of them here, being a bit cheap. And these affect the production of the protein via mutations which simply stop translation at a very early stage and no protein is produced. They affect the processing of the protein in class 2, and the most obvious example here is delta F508, by far the most common mutation. And in class 2 mutations we have misfolding of the protein and then degradation of it. Class 3 mutations, where we have errors in regulation, and an example there is G551D, where protein gets to the surface but has no activity once it gets there. Then we have mutations in class 4, 5 and 6 that are associated with slightly less severe lung disease, where you will have the presence of either mutations which affect the activity of the protein electrically or its levels of abundance. So we have some functional protein getting to the surface of cells. So we have a spectrum of severity of disease loosely relating to the type of mutation. It should be said, though, that of the over 1,900 mutations that are associated with the disease, actually relatively few have actually had proper classifications into this structure. It's not a hard and fast thing that you can easily do. Obviously, though, over the last couple of years, the G551D mutation has received tremendous attention because of the licensing and development of the Kaleido drug that's now on the market from Vertex. So I thought it was worth a little bit of time just looking at that. and trying to understand why it's why this has happened. And the interesting thing about the G551D mutation is that it almost makes fully functional CFTR. The protein gets translated, it folds properly, it gets processed, it gets to the membrane. It's just that when it does, there's no ATP binding, and it's a non-functional protein. So what you have is a mutation that only affects the very last point of G551D processing. And so this is a very, very specific issue just for G551D. It only needs a bit of activation and then it will work. And the problem with random drug screening is it's phenomenally expensive, but what Vertex have been able to do by focusing on G551D is they had a high chance of finding a drug that would have specific activity on just that type of mutation. And that's why Kaleido has been so successful, because it targets it's only really required to do quite a minimal thing. Not downplaying it, it's phenomenal, but it's a fantastic development. But why doesn't this work for other mutations? In particular, why doesn't it work for delta F508? But fortunately, with delta F508, it's a much harder mutation to fix. Delta F508 CFTR fails much earlier. It's a mutation which affects the protein folding, and so it gets recognized as it folds wrongly and gets removed from the cell. And unfortunately, we've evolved to have exquisite systems for the removal of unfolded proteins. Even proteins without mutations are prone to folding improperly, so we have to have a system to get rid of them from our cells. So to fix delta F508, you need a drug that can refold it, keep it folded, have it processed properly, have it get to the membrane, and make sure it's active once it gets there. And so because you need something here which targets much more general aspects of cell physiology, you're far more likely to have off-target effects. And this is why it's been much harder to date to find something which can fix delta F508 CFTR. Although with their other drug that's in development, Kaleido are certainly getting some interesting results that we're seeing there too. Because the problem with random drug screening is it really is this question of how much random drug screening you can afford to do. It's a phenomenally expensive process. Genetics is usually very, very subtle, and unfortunately, CFTR is one of the few genes where it's not. It's not that common in genetics to have such a severe effect for a mutation in one single gene. But for those of us who work in gene therapy, this really shouldn't affect us, because all gene therapists really need is a copy of the correct coding sequence, and we can go away and develop what we want. I'm just going to give you an example now of how that's not really always the case. I want to first of all, you can buy virtually any gene sequence nowadays online. So getting a hold of a CFTR coding sequence is trivial. Don't pay that much for it, but you can go online and buy any coding sequence you like. And in our case, we're very lucky because the CFTR coding sequence has been known and been well established since 1989. Or so we thought. I want to take you back to spring 2006. At that time, we just completed about 2 or 3 years' work. We're very happy with ourselves. We had in our hands our plasmid design that would later go on to become our clinical trial plasmid. The plasmid vector had been tested in multiple systems. It was absolutely ready. The CFTR sequence we had was functional, and we had a manufacturing, critically, we had a manufacturing slot booked for this that needed to be filled. Then one day in the spring that year, our roof fell in. Our collaborators in Japan noticed something very odd. The CFTR sequence, the reference sequence that is available online, had changed quite subtly in March of that year. And now, randomly, there were 2 changes to it. At position 620, it seems very, very minimal, but it was suddenly now a histidine amino acid instead of a asparagine. And at position 833, it was now phenylalanine, not leucine. Now, you would imagine that when something like this happens, there's an alarm that goes off in every lab around the world that works on the CFTR gene. That's what things really should be. Unfortunately not. It just happens very quietly in the background, and unless you notice it, and we were very lucky that they noticed this, you would never ever pick this up. And the reason that this happened is that sequencing genes really is a numbers game. It's about how many you've done and how good your system is for doing this. The N620H mutation was simply an error dating back to 1989 that took an awful long time to be noticed. The L833 is a little bit more interesting because the leucine was in the original 1989 sequence, but in the population at large, the phenylalanine is by far the more common variation. Roughly 90% of people have that. So the original sequence that we had, that we'd been working with, was functional. It had normal CFTR electrics, you could visualize CFTR protein on a gel, and it's a sequence that we think has been used in every other CF gene therapy clinical trial to date. So one response that we could have had was simply, let's not bother doing anything, let's not bother changing it. And we were 90% certain that it would make absolutely no difference. But if you're doing a clinical trial, with the amount of money that was committed to that, 90% certain isn't anywhere near enough, and we were on a very, very tight timeline. So what we had to do was come up with a way of fixing this quickly, and for various reasons it was actually relatively difficult to fix. And so we had to come up with a plan, and I had a plan that was risky, cheap and quick, and we had another plan that was more guaranteed, much more expensive and slower. So with the problems that we had and the timelines involved, obviously we had to pick both. We couldn't just rely on one of these plans. And things played out. I think we got halfway there with my risky idea, and then the more expensive one fixed the second mutation. And the result was our final clinical trial plans with PGM169. And here are three differing views of 169: the map, it being produced, and one of our final vials for the clinical trial. So why am I telling you all this? We had to do a little bit more work. Boo-hoo, you might think, and rightly so. But I wanted to exemplify it. I wanted to talk about this because it was a real lesson to us that not all CFTR variations obviously change its function. You can have variation in the sequence and it doesn't really necessarily affect anything. But also the notion, we have notions that genes are, that we have a single fixed sequence for a gene, and that's all that the gene ever is. And in truth that's quite misleading. And also the records that we keep of human sequencing are often going to be prone to human error, the amount of material being collected, amount of information being collected. And it's also an example of why sometimes research can be so expensive. We noticed this issue really early, and we got onto it really quickly, but we were on a tight timeline. So for 6 weeks, as we fixed this, everything else that we were doing in the lab had to be shelved, and we had to spend money on 2 strategies, so we had a backup. Then we were faced with the problem of, well, we've made this new sequence, but there's a real chance that this correct sequence might actually be worse. So we had to send it to London for UTIS people to test it properly in their models. And really only we have the slightest hint in some systems that it might actually be more active in cell culture models. Was any of this worth it? Well, there have been 25 previous gene therapy clinical trials of CF. None of them, we believe, used what turns out to be the most common functional CFTR sequence in the human population. The one that we're using now definitely does. So we have some reassurance there from that angle. I just want to finish now by looking into the future a little bit and think about where genetics research is being driven to. This is the type of machine that was used in the early 1990s to sequence the human genome. Machines like this were used to carry that out. Over a period of about 12 months, hundreds of these machines were used to sequence the human genome at the cost of around about $1 million, many millions of dollars. The follow-up to the Human Genome Project was what very few people actually know about. It was called the 1000 Genome Project. This finished a couple of years ago, and over 1,000 human genomes were sequenced, and the aim was to map every common instance of variation in the human genome. And the modern day equivalent of that sequencer I just showed you is one of these, a machine like this one here. And nowadays these machines exist, and they can sequence an entire genome just on their own in 3 to 4 days for between $1,000 and $10,000. So over the space of about 10-15 years, the amount of material, the amount of sequence information that can be generated has gone up by several log orders, and the amount of material available today is just phenomenal compared to what it was back in the 90s when the human genome was first out. Where is this going though in the long term? We really are looking at using genome sequencing as a real diagnostic tool. There's a company in Oxford who I've got no connection with, Oxford Nanopore Technologies, who are developing a completely revolutionary type of sequencer. These aren't quite on the market yet, but within the next 4 to 5 years they will be available, and they're going to be able to sequence an entire genome, something the size of a human genome, for about $100, and they'll do it overnight. So what this really does have massive implications for genetics. You'll have much earlier diagnosis of diseases. You'll have far greater power to do modifier gene analysis, to be able to find other genes that have an impact on CFT, on cystic fibrosis outcomes, like inflammatory genes or genes involved with nutrition. So this will lead to hopefully better outcome predictions for patients and much smarter clinical trials by being able to cohort patients much better. This is going to be a real transformation of genetics, and one question obviously is in the UK anyway, is our NHS ready for this? And quietly, they really do appear to be. There's a group in Oxford, for example, a couple of floors up from where I work, who are really trying to use this type of next generation sequencer to replace routine microbiological screening with genome sequencing. And the infrastructure for this they already have in place. It's embedded within the hospital, within different departments, and even out into GP surgeries. And what they're working on is to get tight geographical mapping of infections where they break out. In particular, they're interested in stopping or getting ahead of antibiotic resistance infections. And they really are. They are doing this as we speak in Oxford at the moment. It's been incredibly powerful work they're doing. So they're just waiting really for the sequencing really to catch up with them. How will all this affect CF? Well, we're moving into a time over the next 5 to 10 years where routine genome-based sequencing will be carried out on sputum samples and other patient samples, and this will hopefully lead to far better infection control, less infections, and will reduce the spread or the development of antibiotic-resistant strains. So, I haven't really been able to cover everything massively in depth today, but there's tremendous resources online if you want to learn more about CF genetics, particularly from the cystic fibrosis mutations database that's run by the Sick Kids Hospital in Toronto. And scientists always say that something revolutionary is just around the corner, and I realise I've been very guilty of saying this. But in terms of technology that's driving genetics, it really is true at the moment. And over the next few years, we're going to see potentially huge benefits that come out of this technology, and hopefully a lot of that will be applied to better outcomes for cystic fibrosis patients. Thank you very much.